Welcome, everyone. I think we'll get started. It's my pleasure to introduce Dr. Khanna today as our RIDE seminar speaker. Dr. Khanna is the founder and board chair of Ethos Discovery, a nonprofit incubator focused on advancing scientific innovation to address complex medical challenges and enhance patient outcomes. He's a graduate of the Western College of Vet Med and holds specialty training in veterinary internal medicine and oncology from the University of Guelph and the University of Minnesota, respectively. Dr. Khanna is a diplomat and past president of the American College of Veterinary Internal Medicine, Oncology. He also earned his PhD in pathobiology from the University of Minnesota and was recently awarded honorary membership as a diplomat of the American College of Veterinary Pathology. Following his postdoc fellowship, Dr. Khanna continued his work at the National Cancer Institute as the head of the Pediatric Oncology Branch's Tumor and Metastasis Biology section and founding director of the Center for Cancer Research Comparative Oncology Program. In 2011, he was granted full tenure and promoted to the position of senior investigator at the National Cancer Institute. He's received the NCI Award for Outstanding Research and the NCI Distinguished Mentor Award twice. And he has over 100 publications in the area of cancer biology and therapy. Despite all these roles throughout his professional career, Dr. Khanna self-identifies as a clinician first. He was an active clinician within his referral oncology practices, the oncology service based in Greater Washington, D.C., until 2021. He's also the founder of Animal Clinical Investigation, a contract research company involved in complex medical problems seen in pets. It's my pleasure to welcome back Dr. Khanna.
Thank you very much. Really great honor to be here. Thank you to the organizers and to the college for hosting this event. It's really a great honor. And with the generous introduction from Dr. Snyder and the time we have left, I'm happy to take any questions. We joked, so Amy and I go way back, as do I with Lindsay and Ford in the front. And so I'll pretend that we're back in a rounds room, maybe not as inappropriate as we were then now, and we'll just spitball this together. But the opportunity to share this example of what I saw in the 3-minute thesis is really exciting to me. So the examples that I saw in the 3-minute theses presentations today were exciting because they're examples of how valued and valuable this comparative approach to life sciences problems research is. And I will share with you my example, which won't be 3 minutes, but it's about metastasis in cancer, and specifically in osteosarcoma, and the work we've done to try to resolve an understanding and bring better therapeutic outcomes to human and animal patients with this disease using this comparative approach. Amy referenced most of the information on this slide, but as a result of my being Canadian, I was programmed to retire at 55, which was last year. And so as a result of that, I have no really important financial disclosures to share. One that I will discuss is a relationship with a biotech company focused on metastasis drug development called Vujade. Vujade is the opposite of deja vu. I have nothing to do with the name, but the idea is that this company seeks to prevent the deja vu moment of a physician telling a patient that the cancer has come back. And so I serve on the board, so that's really my only disclosure. My work that Amy described within this incubator of scientific innovation, Ethos Discovery, is a not-for-profit, and the perspectives come from my work as a research scientist within the NIH and a clinician within my practices in the DC area. And that balance, which is almost always equally shared, is sort of why I think the way I do. And so it's a disclosure and a warning for all of you. And as I see Mike Henson there, I'm also reminded that something that Ford and Amy and Mike and I talked about is in the olden days when you didn't know the answer to a question, the answer was either Cocoa Puffs or ultrasound. And it's probably still Cocoa Puffs, but you can replace it with RNA scope. So if that's sort of the hint as we move forward through the discussion. So as a continuation of this disclosure, this for-profit business is what I retired from. I no longer have an association with, but it is a national group of veterinary specialty and emergency practices across the US and Canada, the result of which is we are able to do clinical research within these practices. The basic biology starts in the lab and then matures and evolves into the clinical trials that I'll discuss through the presentation today. So the core of the talk is really what you heard in the three-minute presentations, the thesis presentations, and this is the idea that we, all of you, as a product of being in this summit, are exposed to this comparative approach to life sciences problems. And in that comparative approach, what I think this means is we recognize the most important problem, and we then figure out how to answer it. And that is distinct from the alternative, much more safe and common approach, which is to think about what you do well and do it again and again and again, irrespective of what the problem really is. And that happens to be, and I'm not naming a profession or a discipline, that happens to be the alternative to this approach that we have, and that you're all sort of, you may not recognize that you have this, but you do. And when I think about it for myself, when I started in vet school, I was sitting beside a woman who studied feedlot pneumonia, Haemophilus pneumonia in feedlot cows, and a guy who studied feline asthma. And totally different problems, but we were trained in a process, as a Canadian, we were trained in a process to understand, to know that we could figure this out. We didn't know how we were going to figure it out, but there's a process by which you would do it, and the confidence engendered in you to go about and solve that. And that is very distinct from the approach that says that I know how to do X, and I'm going to apply X to Haemophilus pneumonia, and that's all I'm going to do. And I'll do X to asthma, and I'll do X to PRRS virus, and so on. That X approach is the alternative. It is less valuable, and therefore you are more valuable and valued in life sciences research, and that is really all I need to say. Everything else I'm going to talk about is my example of living that in primarily the human life sciences research environment. So I'll talk about the problem of metastasis very briefly, talk about the one protein that I've studied. I'll tell you the secret about why I study it since we're a small group, and then how we evolved that biological understanding of a protein into an approach to translate biology to therapy, do what we call comparative oncology. And when I was at the National Cancer Institute, I was part of the team that founded the National Cancer Institute's comparative oncology program, which answers questions in dogs with cancer that you can't answer in conventional preclinical models, you can't answer in human clinical trials, and you make better progress, or so the hypothesis goes, than you would if you were left to guess without those answers. And then I'm going to move on to the future. And so the icons here says sort of where I did this work. So more or less academic life sciences research at the National Cancer Institute, evolving to clinical trials with Ethos Discovery now. And the last step will be the work at Ethos Discovery, a human not-for-profit group called the Osteosarcoma Institute, and this biotech company, which I gave you the disclosure for, called Vujade, where we're going to iterate and solve the problems that you will be given a hint to as I walk through the talk, and do what I call Comparative Oncology 2.0, which is really a charge to all of you. Any of you who are interested in pursuing this work, Comparative Oncology 2.0 represents what I think the future of this comparative approach to cancer research will involve, and a representation of the hard work that's yet to come. So Kaplan-Meier survival curves are used to summarize outcomes in cancer, and you can see these curves representing a decline, meaning a smaller and smaller proportion of patients alive, until you get to a plateau, which becomes their survivorship. So in osteosarcoma, children, like dogs, present with appendicular primary tumors. They're cured with surgery. That primary tumor is cured with surgery, no problems there. No additional needs really needed for that presenting problem. But the cancer spread to the lungs, almost always. And the spread to the lungs is the problem for patients. And prior to the era of giving chemotherapy to patients, less than 20% of these patients survived. And you can see most of the patients died within the first 3 to 4 years. So that's a devastating disease represented by the era before chemotherapy. Chemotherapy in the 60s showed an improvement in survivorship. And really over the last now 40 years, no significant improvement in outcome after combination chemotherapy was introduced. And so it's unlikely that giving more cytotoxic drugs that target rapidly dividing cells or induce apoptosis is going to solve this problem. And so we and others would argue that understanding the biology that underpins the ability of cancer cells to spread from the bone to the lung would uncover targets that you could therapeutically muck around with and deliver better outcomes. So that was the goal behind the research, is to deliver more favorable outcomes than we've seen in the last 40 years or so in children. The same story in dogs. And Mike and I are treating dogs with osteosarcoma the same ways we did back in the day, and same outcomes really, with really no change. And the problem in dogs within a group of friends can be characterized by this cartoon, and probably a more aggressive disease in dogs with a little bit of hand waving than it is in humans. And that problem in dogs, in my patients, represents an opportunity to solve the problem in dogs and in children, which is the recurring theme of this cross-species, solve the problem in the best way you can sort of discussion. So to talk about the biology of metastasis briefly, a primary tumor forms really for two reasons, and that's that cells develop uncontrolled proliferation, or they develop a mechanism to resist apoptosis, and that's all that you need to form a primary tumor. Very few patients die of primary tumors, and a number 12, number 10 scalpel blade will solve that problem for the most part. But after that, all of these biological steps occur that result in metastasis, and all of that biology lends itself as an opportunity to solve biology and uncover therapeutic opportunities, which has been our focus. So to summarize that, after the primary tumor forms, cells need to move, create blood vessels, or co-opt existing vessels, and the cells need to get into the circulation where they resist the process of cell death associated with loss of cell contact. So that process of anoikis is a new thing that the cell needs to overcome. Through the entire process, they need to evade the immune system. Almost every cell that forms a primary tumor can do all of this and can get to the secondary site where they physically just plug progressively smaller and smaller vascular structures. They just physically get stuck. There's no biological intricacy to that. And most of the cells can do that. Most of the cells can squeeze their way out of the vessels at the secondary site. In the osteosarcoma example, that's the lung. But very, very few can survive at that secondary site. So you can imagine these cells were programmed to live in bone. And as we heard in one of the thesis discussions today, the bone microenvironment is remodeling constantly, but it's a unique environment, and these cells all of a sudden find themselves in the lung. And so they have to detect stresses that are associated with that new microenvironment. They detect the stress, adapt, and survive, and that's the minority of cells. Most of the cells that get through this process die at that point, so when they're experiencing the stress at that secondary site. So I'm hinting to the fact that biology has delivered us an Achilles heel, which is survival at that secondary site. And so why not take advantage of that Achilles heel and try to muck around with that biology rather than all of the rest of the steps which almost every cell can do pretty effectively, or that's the argument that we try to make. The cells that do survive either form extravascular metastases or intravascular metastases. They grow through the same biologies of proliferation and resistance of apoptosis to produce the visible lesions that then kill the patient. And that's the biology of metastasis that we seek to understand. And some interesting ideas in this process. When I talk about the fact that I study metastasis, people think I study the process, which means the verb, that is the word metastasis, but it also is the noun, which is the lesion, which exists at a secondary site. And I study both because they both uncover different targets for therapeutic intervention. So it's important to think of both of this as a verb and a noun. The second thing to say is that if you think about metastasis and if you think of this clinically, many people would say that the cells have left the bone, they're sitting in the lung, and mucking around with this biology it's irrelevant because it's too late. The horse has already left the barn, and that's the analogy, but it's probably untrue. And that's because the cells leave the primary tumor and fall into a sanctuary site, like the bone marrow. And the sanctuary site is a safe microenvironment where stem cells can live for centuries, literally. And metastatic cells probably sit in sanctuary sites. And then for some unknown reason, they break dormancy and then go to the lung. So that means even after the patient comes to see you, all of these steps have yet to occur, and you can intervene in those biologies and seek to deliver better outcomes to patients. And the last thing to say is that even after a patient presents with metastasis and comes to see you, the process still continues because metastases metastasize. So it's never too late to look at these as opportunities to intervene. And I'm playing on that because I'm counting on the fact that you'll allow me to study this Achilles heel as an argument. So this is the front end of all of our grants, because the criticism is it's too late, you've missed the boat, this is a waste of time. And so I'm focusing here to convince you that it's not a waste of time. And there's reason to believe that. So this is a plug for a textbook that we've just finished editing, and the biology of metastasis is summarized here. Nor I think will anyone profit from this book. But there's some good chapters by some really advanced thinkers on the topic of metastasis and metastasis therapy, as well as other topics related to oncology more generally. So I'm going to move on from the problem of metastasis now to the biology of metastasis and our efforts to find this protein called esrin. And esrin, I'll get into the digression briefly, has been the focus of my scientific career, and it lends itself to translation, and I'll talk about that briefly now. So this comparative approach recognizes that all models are flawed, and it's up to the investigator to identify the question that's best applied to a specific model and recognize the weaknesses of that model before you interpret the answers and assume that the model failed or that the concept failed. So taking this approach, we started my first paper as a fellow with a mouse model of osteosarcoma that's characterized by metastatic variants that differ only in their metastatic proclivity. They're on plastic, they proliferate identically. They're on plastic, they die at the same rate. They form primary tumors in mice at the same level, but one variant is highly metastatic. The second variant is non-metastatic. The highly metastatic variant is called K7M2. The non-metastatic variant is K12. None of that is really important. At the time, we had this breakthrough idea, and this is going to be comical for some of you. We had a 4K cDNA array in the mouse. And so this was a huge opportunity to subtract differences in gene expression between these two models to find what was underpinning the metastatic proclivity of the highly metastatic version. And so we uncovered esrin. Our villain 2 is the gene, or the transcript, which encodes for the protein esrin. And you can see esrin in the highly metastatic cells. So protein validation here by in situ immunofluorescence. The highly metastatic cells having high esrin at the cell membrane. the low metastatic cells having much lower levels in general and not at the cell membrane. Ezrin is a band 4.1 superfamily protein. It links the actin cytoskeleton to the cell membrane at the cell membrane itself. That linkage allows the cell to physically engage the microenvironment and functionally allow signaling from a variety of cell membrane-based receptors. This paper attempts to argue that out of the 100 or so differentially expressed genes, there was a biological reason to select Ezrin. And you can read that paper and take it for what it's worth, but since I'm with Amy and Ford and Lindsay here, I'll tell you the truth. So I did want to do my internship at Cornell University. I didn't get into Cornell, so I had a chip on my shoulder. So Ezrin was named after the founder of Cornell University, Ezra Cornell, and that's why I studied Ezrin. It had nothing to do with this. It was just because I was so upset that I didn't get into Cornell. And that seemed like a bad day, but it turned out not to be a bad day because I went to Guelph, and then Ford convinced everybody here to let me in here. And I met my wife, and everything went well. So that wasn't a bad day at all. That was actually quite a good day. And it led me to Ezrin. So thank you, Ford, and thank you to Ezra Cornell. So we did the obvious, which is to inhibit Ezrin in the highly metastatic cells where the mice died of metastasis. And as we roll back Ezrin, we roll back metastatic proclivity in the mouse model. And so that's relatively easy to do. The gene isn't important in a mouse model of cancer. But how do you instill the confidence in your program to spend 2 decades studying a protein? So the way we did it, I was answering the same question in dogs using a tissue array in which we had hundreds of dog osteosarcoma samples on a single slide. And we could ask the question, how does Ezrin expression in the primary tumor impact survival in dogs with osteosarcoma? And in a matter of hours, and probably literally weeks, we could answer that question. And high Ezrin expression, like in the mouse, predicted a more metastatic progression of metastasis to the lungs. 3 years later, much less robust example of mature data, we could answer the same question in children. And the same result, but you can see much less robust, smaller numbers, not as significant as what we saw in the dog. But this result gave us the confidence to stick with this for 3 years. And it allowed me to sort of tread water through tenure. And we could show that there's something about Ezrin. And we stuck with it through this comparative approach. Now, we could have just studied Ezrin alone, but we then asked, what is Ezrin doing in this complex cascade of metastasis? And using a functional analysis of the Ezrin phenotype, we found that Ezrin was associated with the process of protein translation and the initiation of protein translation, which was surprising for a protein that linked the actin cytoskeleton to the cell membrane. And we found the same thing by a different approach using proteomics when we eluted the proteins that were bound to Ezrin. And again, the function of the proteins that bound to Ezrin were functions of protein translation and the initiation of protein translation. And so this leads to a hypothesis that Ezrin is influencing metastasis by allowing cells to produce proteins, and presumably the proteins that they need when they're stressed on time. And this then takes me back again to the concept of unfolded proteins that we learned about in host response to bacterial infection. And the idea was joyriding pigs. So you get pigs that are happy and healthy, you put them in a truck, you ride them around, they get stressed, and all of a sudden they're sick. So the idea was that the ability of the host to respond to a stress is dependent on these highly structured mRNAs that are kept around as mRNAs, and they sit there waiting to be turned on quickly, not only in time, but in the right place in the cell to protect the host from infection, as I described, but to allow the cell to respond to stresses, as in the case of oncogenes and tumor suppressor genes and genes associated with metastasis. That's the hypothesis. And so we believe that there was a reason to think about how Ezrin was regulating these weakly translated proteins that share in common structural complexity in their 5' and 3' UTRs. So the hypothesis is that efficient translation of proteins that you need, when you need them, in the place you need them, is how Ezrin gets cells over that stressful new microenvironment and allows them to survive. Those cells survive and go on to kill the patient. When you don't have Ezrin, you don't adapt, you don't detect stress, and those cells die off and they never cause a problem. So with that in mind, we started to wonder, how can we model this outside of the mouse? And a veterinary surgeon in my lab came up with this model where we deliver green fluorescent cells to a mouse, either by orthotopic injection to the leg or tail vein injection. And these green, we then sacrifice the mouse, insufflate the lung with agarose, bread loaf section the lung, and put it on surgical gel foam, which was his idea, which keeps the lung at an air-fluid interface over 30 days, and allows us to then watch the process of metastasis in a structurally intact lung over 30 days under a confocal microscope. And so you can imagine we have lung collagen by second harmonic imaging with green tumor cells progressing over 30 days, and we can watch the process with and without Ezrin, with drugs sprinkled on top or not. And that's what allowed us to sort of dig into the biology here. And so you can see the highly metastatic cells with high Ezrin producing what you might call a metastatic lesion, and the low metastatic cells with low Ezrin getting there, but then dying off and never forming an actual lesion. So that sort of continues to conceptualize this hypothesis. And so because we had this model to study these early Achilles heel concepts in metastasis, many labs at the NCI who were around us came to us and said, study our genes, study our drug, and let's look into this. And so what we found is in 10 out of 11 instances, it was all related to survival at the secondary site. That's what a gene or a drug did. Even though these other steps in metastasis are important, there's a dependency on survival at the secondary site. And so these data across cancer types and genes fulfilled the hypothesis there's something about getting over stress, survival, related to successful metastasis, which is what this concept of stages of vulnerability is about. And if you're developing drugs, as I said earlier, why not develop drugs that target an existing vulnerability rather than developing drugs for a biology that's not necessary for the process? So the idea of targeting dependencies is what comes forward. And I will just quickly summarize several hypotheses. Most of this is published, and I'll share with you some of the unpublished. So the first idea is this idea of producing proteins where you need them, when you need them, which I've referenced before. And that work is what I'll summarize in further detail. Another hypothesis, it's only about energy. And if you can deliver ATP to a cell that's stressed efficiently, regardless of substrate, that cell with energy survives, and the cell without energy dies. And so this lends itself to glucose metabolism and ATP utilization. And so that's another hypothesis that I won't get into, but exists. And the last one, which is very interesting, that ties itself to an area of active drug development in oncology, is what's called the unfolded protein response. So it's initiated by the ER, and the sensor of in the cell of stress is the ER, and highly metastatic cells detect and respond to stress in the ER differently, as characterized by their unfolded protein response, than non-metastatic cells. Again, these are both cancer cells, just one is metastatic, one is not. So there's something about the ER and the unfolded protein response that could lend itself to some therapeutic intervention. Again, these are published, and I won't refer back to them in any more detail, except that this has grown as an area of interest because of drugs being developed for oncology for other reasons. So the experiment then to determine if there's really any truth behind this association, any causality behind this association to the unfolded protein response, is to produce a synthetic complex 5' UTR upstream of luciferase and transfect it into cells with high Ezrin in black compared to low Ezrin with red. And you can see that the cells with high Ezrin, which are highly metastatic, can melt through that complexity and report the reporter. and the cells with low ezrin cannot do it. So it suggests that this hypothesis is supported by this model synthetically weakly translated protein transfected into these mouse osteosarcoma cells. When we do the same experiment across species, now dog and human, using siRNA approaches, whenever we knock down ezrin, we knock down the ability of the cell to melt through that complexity and report on this weakly translated protein. So there's further support for this hypothesis. And I'll now transition from the biology into how do we turn this into something that's druggable. And that begins by realizing that the initiation of protein translation and translation itself is mostly mediated and controlled by mTOR. And the downstream consequences of mTOR signaling are the new proteins that are produced by the cell that needs these proteins to survive stress. And so that allows us to then target mTOR with a commercially available and approved drug, rapamycin, and its analogs, the rapalogs, which target mTOR itself, and that's where they got their name. And we can then sprinkle rapamycin or the analogs onto these culture systems. And you can see the untreated lung here, growing metastatic lesion. And this is 10 nanomolar rapamycin. So you could put this in drinking water and not harm the host. And you can see that shuts down the ability of the cells to produce these lesions. And so there's something about rapamycin that's interfering with this ability of these cells to adapt to stress. And so we did the pharmacokinetic studies in mice. We did the pharmacodynamic studies in the same model to show that the drug exposures of rapamycin that are achieved in mice are sufficient to turn off the protein translation. And we just use a different color reporter to visualize this pharmacodynamic event in mice. So these are check boxes as you're developing a drug. Can you give enough of the drug? Does the drug that you give do what you intended to do biologically? And the answer in this mouse model seems to be yes. Sorry, in this ex vivo model and mouse model appears to be yes. So that allows us to then continue the process into mouse experiments itself. And so now across mouse models, whether they're tail vein models or spontaneous metastasis models, all transplantable models. None of these are genetically engineered mice. All of these are transplantable models. The answer appears to be, again, that giving rapamycin turns off or shuts down some part of metastasis. So it gives us the confidence to grow this biological to therapy translation to a comparative oncology dogs study or set of studies. And this idea stems from the basic notion that most of us look more like our pet dogs than we do like mice. And that's true in the cartoon, and this is a British pet food commercial. And although that I think is true at the cartoon level, as a cancer biologist, the cell-to-cell differences in heterogeneity of the tumor yield the problem of cancer resistance, recurrence, and metastasis, which is the essence of the problem of cancer, modeled in dogs spontaneously, naturally. Very difficult to reproduce in mice. We have to really stretch the systems to do this effectively in mice. So these are the reasons that I think that the dog are most valuable for translation and why the mouse models fail alone. And all of these other are potentially additional reasons that the dog can be useful. And Melissa Paoloni did this experiment using, again, transcriptional array study that shows that you can distinguish normal tissues from tumor tissues, but you can't distinguish, and I'm sorry that the colors don't show up, but you don't have a cohort of dog and a cohort of human osteosarcoma. So across thousands of orthologs, the dog and the human are intermixed, and you can't distinguish them. So this doesn't say that the dog is a good or a bad model. It just says that using the dog as a model is a reasonable hypothesis. So that hypothesis underpins the ongoing questions that we ask. And this idea has been in place for a long time. Rainer Storb at the Hutch used dogs with lymphoma to develop preparatory regimens for bone marrow transplantation in the 60s. We've had a lot of milestones along the way, including a national mechanism to deliver on clinical trials, of which the University of Minnesota is a part, led by Amy LeBlanc now through the NCI's Comparative Oncology program, a genome, and this is a typo that I need to fix. Anybody who's a regulatory person and the veterinary pathologist in the room is already getting upregulated now, because there's not regulatory guidance, there's regulatory clarity. Guidance means a lot to veterinary pathologists, and I don't want to get them upset. There's not regulatory guidance, there's just clarity. Okay, so all of that allows us to do this now at scale. And so at scale means a national capacity to do clinical trials, resources and reagents to ask why drugs work and why they don't work, and a mechanism to advocate within the community on when these dog trials are going to be useful and when they're not going to be useful. And that's increasingly an important part of the discussion as there's been traction around this hypothesis. We had a meeting to ask in osteosarcoma, how do you develop a drug that uniquely targets metastasis but doesn't target the primary tumor? Okay, and so you can imagine that early phase clinical trials in humans and in dogs ask if a tumor shrinks. Okay, but if you don't target proliferation or apoptosis, tumors don't shrink. And if the goal is to develop a drug that prevents cancer cells from spreading, it only occurs in late clinical trials, meaning $8 million later, that you can measure an anti-metastatic effect. So then I have to go to someone like Ford and say, I have no evidence that this drug works in any species, and especially in humans, and it does make tumors smaller, but I want you to give me $8 million, and we're going to spend 10 years asking for the first time, does this drug work? And usually he says no. So this paper was intended to address Ford Bell, and all of these people agreed that the dog is the answer to solve this problem. So dog clinical trials in a short period of time, preventing metastasis, can give us the answers about drugs that prevent metastatic progression. So this includes people from the FDA, experts in childhood cancer care and veterinary oncology. And so they all agreed, and they all get their name on it, and they allow me to say that they agreed to this idea. So I've saved you the time of reading this paper. And so the high idea was you can do one trial in humans, and it takes 8 years to do one trial in humans internationally to ask if a drug works. And so every 8 years, you better have a pretty good idea. Otherwise, you've wasted 8 years, and these children are dying of their disease. So why not take advantage of the fact that we have 10 to 100 times as many dogs with this very similar cancer, and pick a winning drug. So pick a winning drug in a dog trial, and then move it into a human clinical trial on a timeline that suits that 8-year cycle. And so that was the idea, and it turned out that rapamycin and the Rapalog was ready for this translation. There were some problems, and I'll get into that, but there was also, as courtesy of Dr. LeBlanc, who now directs the Comparative Oncology program, she was able to initiate broader traction and input from the animal health community, so you can see real marketing dollars and not me making PowerPoint slides anymore, behind an effort like this, and then partnering with the pediatric oncology advocacy community to do this type of cross-species research. And so the questions that we would ask to design the best human clinical trials are listed here. And you can see that for the most part, we're not asking, does the drug work? We're asking questions that would prevent making a mistake by moving a drug forward that's not going to work. That's how we're picking the winner, by culling off the ones that don't work. That's more of the agenda and the idea, because the drug is going to have to succeed or fail in a phase 3 human clinical trial anyway. So we're not going to try to overtake that result by doing something in the dog itself. On the PK/PD issue, I'll sort of give away a problem. So we knew what drug exposures were necessary to give rapamycin as an organ transplant drug, and so we modeled that PK for these dog studies, but we didn't know what PK and PD was necessary for an anti-metastatic drug. And we had the option to develop that knowledge and then convince MD physicians to use the drug that they're used to using in a different way, or to just use the drug they're used to using in the way they're used to using it. And getting MD physicians to change what they do is really hard, even harder than getting veterinary clinicians to do it. So we chose to take the path of least resistance and model the exposures for dogs around organ transplantation uses of rapamycin, not anti-metastatic uses. And that's a hint to say that was maybe a problem. We confirmed chronic tolerability, and then we went on into a prospective randomized nationwide clinical trial led by Dr. LeBlanc. And the punchline is it didn't work. So you can see these survival curves completely overlapping, and we're left with, it didn't work. So after all of this, I wasted 40 minutes of your life here with something that didn't work. And we're left to say, now what do we do? And an inclination in drug development is throw up your hands and say, target or drug didn't work, let's move on. And that would then throw away a lot of effort. And so we are of the mind that iteration is probably a better idea. So let's dig into what went wrong and can we tighten the screws through iteration. And so this green circle is the exposure to rapamycin that the dogs got in this clinical trial. And they got that narrow window because we made the mistake of modeling it under organ transplant PK, which is not tolerable in combination with chemotherapy chronically. And so they could only tolerate it for that window, so that's all they got. And you can guess that we believe that was a mistake, right? So one question is, should we give it longer? Should we give it earlier? We therefore need a new formulation of rapamycin to be developed as an anti-metastatic drug. And then what are the next questions? One option is to just give up and say, let's move on, and I'll pass the torch off to you, and you spend the next 30 years of your life doing this, and you get to, when you add more blonde to your hair, you'll get to tell the story. But we chose not to do that, and we're in this phase of iteration. And so there are a couple of things we've done. One is we've developed a drug called Rapaboil. And so my business partner is a guy named Ira Gordon. And Ira Gordon is I can't remember the movie so he's the savant of acronyms. So if I was to say, we have developed a Rain Man, he's the Rain Man of acronyms. So if I type an email that says acronym ? And I say, we have developed a new formulation of rapamycin, which is a blend of oils, he sends back in an email almost immediately, Rapaboil. And he does this over and over again. So if you see any acronyms, this is courtesy of this guy, brilliant guy, characterized by his ability to be the Rain Man of acronyms. So one option to iterate is a new formulation of rapamycin, Rapaboil. Did I get that? Oh, go ahead. Well, I'll listen. So that's one step, is new formulation that's been developed, and we can combine with cytotoxic chemotherapy, and dogs tolerate it very well. My dog did not, unfortunately, but most dogs tolerate it very well. Step 2 was to speed up the cycle of innovation. Am I out of time? Okay. I can stop if we need to. Okay, so that last clinical trial, if you can imagine, there's a cohort of physicians waiting for results from a dog trial, and they have an 8-year cycle to make decisions. We can't take 3 years to give them a decision, because lining up 3 years on a clock with 8 years on a clock makes us miss way too many times, and they're just going to say, forget it, I'm just going to move on, right? So we have to speed up the cycle of iteration. And so the second thing we've done is a clinical trial in osteosarcoma called MIMIC. MIMIC is minimally invasive metastectomy in canines. And so what MIMIC allows us to do as an outpatient procedure in large, giant breed dogs that are old is, rather than split their sternum and have them spend 2 weeks in ICU recovering after we remove lung nodules, they have an outpatient-like discharge with 3 pencil hole incisions in their chest and a video scope to extract and remove these lung nodules. So removing the lung nodules, one, may deliver therapeutic benefit, which is valuable to our patients, like it is in humans. Two, it can allow us to then add a drug of interest and expect a much more rapid cycle of learning to learn if the drug prevents new lung nodules from forming. And we can learn that more quickly. And we'll develop new drugs for humans and develop new drugs for dogs more quickly. So this becomes step 2 in the iteration, applying new drugs to this model rather than remove the primary tumor and wait for them to develop lung nodules. So Mimic is now open at one of our Ethos hospitals, sorry, 2 of our Ethos hospitals that have this skill set and the Ontario Veterinary College, and we're recruiting new patients. Mimic also allows us to collect valuable biospecimens to help us understand biomarkers of metastatic progression, burden of disease, using cell-free tumor DNA, and the interest of the bioelement of greatest interest is extracellular vesicles. And I don't want to upset anyone by calling them exosomes, because they're not, they're extracellular vesicles. And if anyone's involved in that food fight, I don't understand it, so I just call them extracellular vesicles. So there's biology that's going to be learned from this process as well. And I'll just back up to say that so far we're getting, so depending on when you schedule surgery, if you schedule these surgeries in the morning, it's an outpatient same day of discharge. If you schedule them at the end of the day, it's the next morning, so it becomes 18 hours. So we're now able to get these dogs to walk out. And I think about the numbers of dogs that I gave a new chemotherapy drug to when they developed metastasis. I never saw much that worked. So I probably wasted time and lives of my patients for decades, and now we can cut these nodules out. And for some surprising lesion in humans, after all of this has happened, and you cut out these nodules, a surprising number are cured, which is a curiosity to me. I don't know why that is, but they are. We'll learn how many dogs are cured, but we'll then learn what drugs to add after surgery to accelerate the cycle of innovation. So this is the Osteosarcoma Institute. We are working with them, working towards a cross-species collaboration so they identify the drugs that they want to test. And I've made this mistake twice, and I'm almost going to make the third time, where I come up with a great idea, I test it, and then I have to find a place to stand to tell physicians that this is a great idea, you got to give this to your patients, and it never works. So the alternative is for them to tell us what to study, and I'm sure that they can convince me of something that they believe is important, but then we have people waiting around, waiting for the answer, and they're going to use it. So this is the idea of this collaboration with the Osteosarcoma Institute. The slide is also intended to remind me that they fund, I chair the Strategic Advisory Board, and we fund trials, studies, biology studies in comparative oncology and osteosarcoma. So think of them for grants in osteosarcoma research. Vujade, I told you about them a little bit. So this is step 3. So Vujade has internalized a series of assays of metastasis proclivity, including the one that I showed you. And the idea is that no drug by itself has ever cured a patient with cancer. It's always a combination of drugs. So how do we find another drug? And so I'm just telling you that we found another drug to mix with rapamycin. I can't tell you what the drug is, just different mechanism, and the idea is 1 + 1 maybe equals 3. And so that's what that is. So those are the steps in iteration that lead us to the final piece, which I'll conclude quickly. And that is that the field of comparative oncology has a national capacity to do trials, regulatory clarity, reagents, endorsement from academia, the federal government, and biopharma. And we could say that let's just wait and have a few more chances, and eventually something will be a grand slam. This is a Charlie Cleasel reference. Charlie Cleasel was a cat who somehow I decided was Amy's husband's nickname. He was a friend of mine. I don't know the story, but I think it was Charlie was the cat. David was someone. It was David. Pam David was this. So Pam David is the vet student. And anyway, never mind. Charlie Cleasel. Sorry about that. I had to get Charlie Cleasel in somehow. So we could wait and just try again, or we could change comparative oncology from an academic exercise into a commercial exercise. And so this is comparative oncology 2.0, is to align biopharma in human health and animal health to make comparative oncology a commercial exercise as a means to increase the opportunities and create what we would describe as a self-funded model. So this paper is published, and it's very simple. Animal health, or so human health biopharma licenses the rights to a drug to animal health, and that transaction delivers funds to do comparative oncology. Right? And so that's been done, but it's not been done enough. One thing we did to increase the size of that transaction is align the FDA on the human and the veterinary side. I won't go into the details, but we were able to get that alignment between the veterinary FDA and the human FDA on manufacturing principles, the result of which that licensing agreement is now worth more, to the tune of $3 million per transaction more. That gives more money to fund comparative oncology research. So that basically is a commercial strategy to fund and self-fund comparative oncology research. Last slide is to say that for me, this idea of us looking more like dogs than mice was at its peak of inflated expectations when we founded the comparative oncology program with the NCI. And after that occurs, not surprised, like this is a figure for any marketing innovation, that later adopters are going to come in and eventually we'll have some disappointments. And these disappointments are going to be because accrual to trials is slow, the wrong questions were articulated in the clinical trial, and the efforts weren't integrated with the human activities. And they will lead to disappointments, and I ask you all to dig in and figure this out so we can raise it to a point of productivity and do this properly at scale. So we've done it as a N of 1, and we now need to do it at scale. And that's your challenge. And it's a huge challenge because this is going to happen if we don't sort of tighten the screws and fix this. So that's really everything I wanted to say for the discussion, but because Ned's in the room, I thought that there may be some DVM PhD students in this audience, and for them, what I would always tell them is your career as a clinician scientist is going to require you to develop more skills as a clinician because your DVM training alone is not enough. And all this slide is to tell you is there's a mechanism to do that without having to go and get a residency and become board certified. It's called ER Immerse. You get paid pretty well to be an ER doctor. You get exposed to all of the clinical disciplines, and then the output is while you're an ER doctor, a few shifts a month, you can find a lab, find a postdoc, and come into a postdoc with real clinical credentials, and you could choose to become a ABVP specialist. And so all of this is to say, if you have an interest, let me know. What I used to tell those people is you got 10 years ahead of you, and that was not fair for me to say. Now this is a 2-year process, and you're on a new track. So just think about this as an alternative to strengthen your clinical credentials before you leave your PhD, sorry, after you finish your PhD. So that's all. So the group at the Comparative Oncology Program, Amy and Christina, who continue to lead that effort, my lab at the Pediatric Oncology Branch at the NCI, who did all of the biology around Ezrin, and my new team within Ethos and Ethos Discovery. So I acknowledge them and thank them and ask you to join us in this effort and follow us as we try to continue this cycle within this not-for-profit. So that's all I have, and happy to take questions.
Well, thank you again.
I was wondering for the second, how is it?
So it's a good question. The second drug targets the same phenotype. So the phenotype is stress adaptation and survival. Second drug targets that same phenotype, but different mechanism, completely different mechanism. And the combination is, so you know the terms synergistic and additive. I couldn't tell you against the phenotype whether it's synergistic or additive, but it's, I'll take the default and call it complementary. Yeah. Thank you.